Alright, so moving on, our next lecture we're going to do is carbohydrates. I have to apologize in advance because my cat is on my lap and he's kind of scratching me, so if I squeal, that's what that's about. Okay, so we're going to talk about carbohydrate structure, how carbohydrates are made, and what they do. But before I get into that, I just want to make a point very analogous to the enzyme ASE point, and that is a lot of carbohydrates end in the suffix OSE or AN, and so if you see again the name of some big long thing you've never heard of but it ends in OS, a lot of times that means, or most of the time that means that's a sugar. If something ends in AN, very frequently that's a carbohydrate. And I don't have it on here, but something with the prefix glyco usually refers to sugar. Okay, so what are carbohydrates? These are sugars. You can have them in a few different flavors. Monosaccharides, which means you have just one individual unit. Polysaccharides, which are polymers of monosaccharides, very analogous to amino acids we were talking about in the last clip. And of course you also have delicious. So carbohydrates functionally have a few very important biological roles, and that involves being structure as part of the cell, and another involves being a storage unit for the cell. I'll talk extremely briefly about the fact that they are also used as building blocks or decorations in either fibrous materials between cells making up a tissue, and they're also important in cell identity. Okay, so the first point I want to make very, very quickly is that monosaccharides, these individual sugar units, are variable in structure, and they vary based on the location of something called the carbonyl group. That's the carbon double bonded to an oxygen. Sugars can be named as a class based on where this group is. An aldose will have it on the end, a ketose will have it in the middle. Sugars can also be named by and categorized by the number of carbons they have. So a triose is a sugar with 3 carbons, a pentose has 5, etc., etc. There's a point to be made here about the spatial arrangement of these atoms and the differing arrangement of where the hydroxyls versus the carbonyl groups are, and the idea that this is going to give different chemical properties. Now, in class, what I actually have people do is count the number of carbons, hydrogens, and oxygens in these two molecules, which look like different sugars, and in fact are different sugars. But what you actually do, if you pause the screen and count them, what you'll find is that these two molecules have the exact same chemical formula, but they have different chemical properties because the same set of atoms are arranged spatially differently. In addition to where the carbonyl group is, you can also have linear sugars and ring sugars, and you can also have some sugars that swap back and forth between being linear and ring. Basically, the way this works is actually simpler than one might guess. This hydroxyl and the carbonyl group will have an interaction. As you might be able to guess, what happens with most of these things is that you wind up spitting out a very innocuous byproduct. Have a guess what it is. Okay, so now we're going to move on to the structure of a polysaccharide, and then the whole rest of this clip we're going to be talking about very specific polysaccharides and what they do in a cell, which I actually think is very cool, even though it seems boring. So polysaccharides are polymers of monosaccharides, very analogous to what we've been talking about with nucleic acids and polypeptides so far, is that you have these big, long, linked up chains of individual subunits. The bond between these individual subunits, last time we were talking about proteins, that was called a peptide bond. Here we call it a glycosidic linkage, but very analogous, works very much the same way. It's a bond between two monosaccharides, and it can happen between any two given hydroxyl groups. Sorry about that. One important thing that's a little bit different from polypeptides is just that these monomers can either be identical or they can be different. With polypeptides, they just tend to be different. It's very rare you'd have a big long peptide all of the same amino acid, but that's very common actually for polysaccharides. I just want to point out here how these glycosidic linkages are made, and hopefully this process is starting to look familiar to you by now, but this is once again a condensation reaction. So you have a hydroxyl here, a hydroxyl here, this is going to link up to this OH and this H are going to be spit out as a water molecule. One other thing to point out is that I have again two examples of disaccharide synthesis. In this case, we're on top we're making a sugar called maltose, and this is where you have two glucose molecules linking together, spitting out water and making maltose. Here, down here we have glucose and fructose, spitting out a water, linking together and making sucrose. So both disaccharide synthesis, the actual linkage happens the same way, it's just in this case we're making a homodimer, and in this case we're making a heterodimer. Okay, so the types of polysaccharides that I think are very important to know fall into two categories: storage and structure. So here we've got storage, down here we've got structure. And there are 5 types of polysaccharides I'm going to discuss. There are 5 specific polysaccharides to discuss. They're starch, glycogen, cellulose, chitin, and peptidoglycan. Starch is made by plants, glycogen is made by animals, and you can see both of these I've marked as storage carbohydrates. The other 3, cellulose made by plants, peptidoglycan made by bacteria, and chitin made by fungi. Some animals for their exoskeleton and even some algae are also is also structural. I bet this is kind of fun to think about, that the idea of the entire exoskeleton that holds a crab together is actually made of a sugar. This is a very busy slide, as is the next one, so I apologize, but what I have here is a diagram of starch and of glycogen to start with. What we've got here is a zoomed in version of any one of the linkages between these individual monomers. What I want to point out is this little grayed out phrase here, which is actually quite important, and we'll get back to at the end of this lecture, and this is that it says alpha 1,4 glycosidic linkage. Alright, that seems rather boring for now, but it's actually extraordinarily important. You'll see why later. You can see glycogen has the same phrase down here, alpha 1,4 glycosidic linkage. You can also see it's got, in many ways, some of the same features as starch up here. When we look down at cellulose, on the other hand, which again is made by plants, we have a different phrase here, beta 1,4 glycosidic linkage. And when you look at the entire molecule, this suddenly looks extremely different from these up here. These are very well ordered, and you can see that they actually seem to line up in these great, nice parallel strands. Starch and glycogen don't do that. They basically tangle up into these big, sort of non-structured balls. One more thing to point out is that we've got these glowing yellow dashed lines here and here. What this is demonstrating to us is that we have this group here and this group here from one strand of cellulose to the next one that are hydrogen bonding to each other. So this glowing yellow line here would be representative of all of these, and this is showing us how one linear polysaccharide molecule of cellulose interacts with a second one and a third one and a fourth one. These guys actually hydrogen bond to each other. This doesn't sound interesting, but I promise it will be. The point of this slide here is that we've now got the same types of images for chitin and peptidoglycan. There are some minor differences between between chitin, peptidoglycan, and cellulose, namely involving these side chains here, but the basic point that I want you to take away is that we once again have these nice parallel structures that are hydrogen bonding to each other, as opposed to the big tangle that we see with starch and glycogen. Okay, so I want to start by talking about carbohydrates that provide structure. So these are again cellulose, chitin, peptidoglycan. I want you to think of the way that these are organized, and they can be organized in either, a nice way to think of either one of them is in ropes or in wraps. So if you see these individual threads of a rope, and each of these were pulled apart and just kind of thrown in a pile, that would be not spectacularly effective at doing much of anything. But when they're nice and structured and organized, they make a rope, which can do something, which holds things together. In addition to that, you can think of it, if it's easier, as a raft. If you had a pile of sticks on the bank, and then you put them all in the river and expected to be able to float down it, that wouldn't work. However, if you line them up and lash them together and organize them and give them rigidity and structure, that will work. It's the same pile of sticks or the same pile of threads, but when you organize them properly, they become functional. One thing that is important to note is that these three carbohydrates that are held together with beta-glycosidic linkages are common and strong and provide good structure because these beta linkages are very hard to degrade. A question to ask yourself before we move to the next slide is, what do you think this means about how essential they may be to the cells that produce them? We actually have a few great anecdotal examples of how structural carbohydrates tend to be indispensable in cells that have evolved to make them. So a great story that comes out of this involves this dude right here. His name's Alexander Fleming. He was a bacteriologist like myself, and much like myself, he was prone to work-related tantrums and messiness. The reason why this is relevant is that he'd done a couple of experiments that hadn't worked as they were supposed to. He got mad, went on vacation, came back, and found plates that looked like this. You see, he found them because he didn't bother to throw them away. Again, much like myself, quite messy. Anyway, what he found was that much of these petri dishes had the Staph aureus bacteria he was trying to grow. Some of them had been contaminated by mold. And what looked odd was that it seemed to be that around the mold there was this nice clear zone where there was nothing growing. So it was almost like the staph bacteria couldn't go interact with the mold. So what was eventually came out of this discovery many years later was that this mold is in fact inhibiting the bacteria by secreting something. This mold is called Penicillium notatum, and so you can probably have a guess that the compound that it secretes that inhibits the staph bacteria is penicillin, which is one of the first known antibiotics. I don't know if anybody had common knowledge of that, but if you've ever taken penicillin, that's where it comes from. In any event, it became clear several years later why this actually kills staph bacteria. And it turns out what it does is that this compound, penicillin, is able to actually clip glycosidic linkages present in peptidoglycan. So it targets these beta-1,4 linkages, cuts that linkage, and takes what should be a nice, strong, solid structural carbohydrate and turns it into individual monomers. Since peptidoglycan makes up a bacterial cell wall, and bacteria tend to rely on their cell walls to hold them together, if you start cutting their wall apart, that's bad, and it makes you become dead. That is why penicillin is an effective antibiotic, but it also happens to be a beautiful natural example of how critically important structural carbohydrates are to the cells that make them. So that's it. I'm going to change gears a little bit and talk about the other kind of carbohydrates we've talked about and make an initial point about carbohydrates and energy, which is how we normally tend to think of them. As a brief introduction to this, nearly everything a cell does is going to cost you some energy. A cell dividing is going to require energy. A cell making new proteins is going to require energy. Enzymes performing their functions fairly frequently require energy. And for now, how I want us to think about it is that individual units of free energy are represented by a molecule called ATP. We'll learn a little bit later how ATP is actually synthesized and why it's such a good shuttle for free energy. But for now, what I want you to know is that individual glucose molecules are the input needed to synthesize ATP, synthesize these units of energy, and again, we need those units of energy to do just about everything that a cell needs to do. Okay, that being said, as a reminder, when we had glycogen up on the slide a few slides back, I was pointing out that it's actually a bunch of individual glucose monomers all tangled up together. On average, it's about 30,000 linked up glucose molecules. So you need glucose again to make your ATP. Starch and glycogen are how plant and animal cells, respectively, hang on to free glucose. So think of starch molecules and glycogen molecules as almost like banks for your individual glucose monomers that you can withdraw from whenever you need to make some free energy and do so. Starch and glycogen molecules tend to be so large that using microscopy you can actually see them. So this is a big ball of starch in a plant cell, and these black specks here are big globs of glycogen in an animal cell. Okay, so how does this work, this transfer of energy? So you have glycogen or starch. They interact with an enzyme. In the case of glycogen, it's a phosphorylase. In the case of starch, it's something called amylase. These enzymes break off individual glucose molecules, so make a withdrawal from the bank, if you like. And then those individual glucose molecules are used to, are converted to generate ATP through a process we'll go through in a few clips from now. The way this works chemically is just that you do the exact inverse of the condensation reaction we saw several slides back. The enzyme attacks this bond, it grabs a water molecule and basically shoves it back into place, takes this oxygen, gives it one of the hydrogens, and then gives the other hydroxyl group left over and sticks it right here. That's it. I have this link to this amylase kind of interactive tool for how this works. There's the link to it. But again, download the PowerPoint slide from Blackboard and click on it. It'd be just as easy. But this is really an excellent interactive tool, and if this is not making a lot of sense to you, how these bonds are broken and how these free units of glucose are liberated, definitely give this a look. It's a very, very, very good interactive tool. Okay, so just to think about this for a minute. Energy, why do we use storage carbohydrates versus structural carbohydrates? Alright, I've already alluded to the answer, but here it is in stark terms. Glycogen and starch are good storage carbohydrates because their glucose-glucose bonds are very easy to hydrolyze. Incidentally, I'm sorry, this reaction right here is called a hydrolysis reaction. So sticking that water molecule in there and breaking this bond apart is very, very easy, and it's not take a lot of energy. This makes some sense if you think about it. If you need to make free energy, you don't want to spend a lot of energy breaking your glucose bonds. You don't want to spend a lot of energy going to the bank and taking out your currency. Another way, if you like to think about it, is if you need some energy and you're hungry, it's much easier to walk into Subway than it is to fight a lion and make some kind of lion steak thing out of it. I don't know, sorry. Anyway, the reason why these glucose-glucose bonds are so easy to break here is because these are alpha linkages. And they're symmetrical. I'll tell you what I mean by that in a second. In contrast, structural carbohydrates, again cellulose, chitin, and peptidoglycan, they resist bond hydrolysis. They are hard to break down, and in fact we saw with the case of bacterial cells with in the presence of penicillin, it's lethal to have them broken down. The reason for that is because they're held together with beta linkages, which are asymmetrical. So what the heck do I mean by that is this. When we were discussing beta linkages and why they came up with those nice parallel sheets, the reason for that was that this group here could hydrogen bond to the carbohydrate molecule in the line next to it. And this group here could bond with the hydroxyl from the group down here. This cannot happen with alpha linkages because these groups are on the same side of the molecule. These are on the opposite side. The point here is that when you have a nice hydrogen bond here and here, this linkage is protected from any enzyme coming in and attacking it. An enzyme coming in from over here can't penetrate between these two bonds, can't get at this linkage, and can't break it. Over here, where you don't have any of this hydrogen bonding, where you have kind of this free-for-all structure, this glycosidic linkage is totally exposed. And so it's very easy for an enzyme and a water molecule to grab onto this and break it apart. Okay? So the big difference between these two, which are seemingly exactly the same monomers linked together, has to do with this beta versus alpha linkage, whether these are on this one side or these are on the opposite side, and that is the whole story. That is the difference between breaking apart easily to make extra energy and being part of an exoskeleton that's going to hold an entire animal together. Okay, so now we're done talking about structure and storage carbohydrates. I want to very briefly mention another role of carbohydrates having to do with something called glycoproteins. So what glycoproteins are, they're proteins that are covalently linked to carbohydrate molecules. You tend to find these in membrane proteins. So this is a cell membrane. We'll talk about that in the next clip. These green things stuck through it are meant to represent membrane proteins. And in each of these cases, these are glycoproteins. This red jazz on the top here is meant to represent polysaccharide carbohydrates. You can see that these are all oriented towards the outside of the cell, and none of them are stuck on the inside of the cell. And here's the reason for that. You can answer it by considering this cupcake. The answer to all of this and the role of glycoproteins has to do with this cupcake. And the way that you can figure this out is, what kind of cupcake is this? So think about it for a second. You can't taste it, you can't smell it, and you didn't make it, so how the heck would you know? And the answer is obvious. It's a peppermint cupcake, and you know because of the sugary things on top of it. So my point here is that a lot of functions involving cell recognition are associated with glycoproteins. So each organ, each cell surface, etc., tends to have a unique sugar signature on it. This is important in cell-to-cell interactions, which I probably won't record for this series, but when I had taught 201 in the past, I tended to speak quite extensively about this. I assume that this is quite important from an anatomical and physiological standpoint, although I'm not quite as familiar with the details on that. But what I can assure you of is that this is extremely important from an infectious disease standpoint. We will talk about that later in the semester, in particular in the context of influenza pandemics. Alright, and that is pretty much all I have to say about carbohydrates. So, lipids next.